A rover on Mars just cracked open a rock. Inside, patterns that shouldn't exist. Minerals arranged in ways that, on Earth, only happen two ways. Microbes feeding on nutrients, or chemistry nobody's ever seen before. For a year, 1,000 scientists tried every explanation. They all failed. Now we're staring at leopard spots 3 billion years old, and the biological answer keeps getting stronger. If you've been curious whether we're alone, the clue might be trapped inside this stone. Hit subscribe if this grabs you. We're diving into the strangest sample ever pulled from another world. Take a breath. This gets wild. July 2024. Perseverance rover grinds across an ancient riverbed on Mars called Neretva Vallis. The mission has been running for 3 years now. The team back on Earth has analysed thousands of rocks, boulders the size of cars, pebbles scattered in the dust, slabs half buried under centuries of sand. Most look identical, reddish brown, weathered smooth, by wind, unremarkable. Then the cameras lock onto something different. An arrowhead-shaped stone, 3 feet long, 2 feet wide, partially buried in a rust-coloured dirt. The rover moves closer. White streaks cut through the rock like frozen lightning. Those streaks are calcium sulfate veins. They only form one way. Water flowing through cracks, dissolving minerals, then evaporating and leaving deposits behind. Water means this place was once habitable. Billions of years ago, this wasn't a dead valley. This was a river, a massive one, 400 meters wide, rushing with enough force to carve through solid bedrock and carry tons of sediment into Jezero Crater. The crater held a lake back then, maybe 100 miles across. Rivers fed it from the highlands, dumping mud and sand into calm water. Where the fast current met the still lake, deltas formed. Fine particles settled. Clay, silt, organic molecules washed down from upstream. Everything life would need if it existed here. The rover's cameras zoom tighter on the rock. That's when Mission Control in California sees them. Dark spots, dozens of them, scattered across the surface like someone splattered paint. Round, distinct, organized in a pattern. They look like leopard spots. Nobody on the science team has seen anything like this on Mars before. Not in 3 years of driving, not in any of the previous missions, not in any meteorite ever studied. The mission controllers face a decision. Keep driving toward the next waypoint on the schedule, or stop everything and investigate this rock. They stop. Because on Earth, leopard spots in sedimentary stone usually mean one thing. Biological activity happened here. Microbes clustering around nutrients, feeding, metabolizing, leaving chemical fingerprints behind when they died. The team needs to know if Mars is telling the same story. Perseverance deploys its drill. The bit spins against the stone. Dust flies into the thin Martian air. The drill pushes deeper, deeper. Then it pulls back with a perfect cylindrical core. Pristine rock from inside, untouched by radiation or weathering. They seal it in a titanium tube. The sample gets a name: Sapphire Canyon. Extracted from a rock they're calling Cheyava Falls, after a waterfall in the Grand Canyon. The rover's instruments immediately start scanning. Chemical sensors activate. Spectrometers fire lasers at the surface. Data streams back to Earth. Organic carbon detected. Sulfur compounds present. Iron phosphate confirmed. Phosphorus, which every living cell needs to build DNA. All the raw ingredients for microbial metabolism or water. Greigite is even rarer. Certain bacteria produce it as metabolic waste when they process sulfur compounds for energy. Finding both minerals together, locked into the same tiny spots, arranged in reaction fronts where chemistry and biology meet, should mean something lived here. Should mean microbes were feeding, breathing, leaving traces in the stone. But Mars has been a frozen wasteland for billions of years. The atmosphere is gone. The water evaporated or froze. life shouldn't be possible now. Maybe not even back then. The team transmits everything to mission control. Then they wait. Because now the real work starts. They need to prove whether life left this signature, or whether Mars somehow learned to fake what biology does. What happens next makes that question far more complicated than anyone expected. Those spots aren't random splashes. They're organized, structured. Under high-resolution imaging, each one shows a bullseye pattern. Dark center, bright halo surrounding it, mineral layers stacked in rings like a tree trunk. Geologists call this a reaction front. It's the boundary line where two different chemical environments collide and interact, where chemistry happens, where elements get rearranged and new minerals form. On Earth, you see these patterns in rocks where groundwater seeped through tiny cracks. The water carries dissolved nutrients, iron, sulfur, phosphorus. When it hits a layer with different chemistry, reactions start. Metals precipitate out of solution. Compounds form. And if microbes are present, they exploit the gradient. They position themselves right at that boundary and harvest energy from the chemical imbalance. They use what scientists call electron transfer reactions, pulling electrons from iron or sulfur to power their metabolism, staying alive in environments that seem completely hostile. As the microbes eat and multiply, they leave waste. Minerals build can form without life. High temperatures can do it. Volcanic heat pushing through rock can create chemical gradients that precipitate minerals in rings. Extreme acidity can do it too. Highly acidic water flowing through cracks can trigger reactions that organize minerals into layers. Even certain organic molecules, if they happen to bind to minerals in specific ways, can catalyze ring formation at lower temperatures. No biology required. So the science team starts running through alternatives. Did these rocks ever get hot enough to create vivianite and greigite together? They check the thermal history. The instruments say no. The mudstone contains clay minerals that break down above 150 degrees Celsius. Those clays are still intact, meaning the rock never got that hot. Did acidic water flow through and trigger the reactions? The team looks for chemical evidence. Minerals that only form in extreme pH environments. They find nothing. The chemistry looks neutral, balanced. Could random organic compounds have catalyzed the reaction without biology? Maybe. It's theoretically possible. But there's no known process on Mars that would arrange them into these perfect little spots. No mechanism to create the organization. The patterns are too regular, too consistent. And then there's the location itself. Cheyava Falls sits in the Bright Angel Formation, a stack of sedimentary layers on the edge of Neretva Vallis. This used to be a river delta, a place where rushing water suddenly slowed down as it entered the lake, where it dropped its sediment load in calm conditions, where clay particles and silt settled gently over millions of years, where organic carbon from upstream accumulated in thick deposits, where nutrients pooled, the exact environment where microbial life could thrive if it existed anywhere on ancient Mars. The team writes everything up, every measurement, every alternative explanation they tested, every reason those alternatives don't quite fit. They submit it to Nature, one of the most prestigious scientific journals in the world. Peer review begins. Other scientists, experts in geochemistry and astrobiology, pick the paper apart looking for mistakes, looking for claims that go beyond what the data supports. They scrutinize every conclusion. 9 months pass, revisions, more tests, more questions. Then the paper gets accepted, published for the world to see. The conclusion is careful, measured. This is a potential biosignature, not proof of life, not confirmation. Just the strongest hint humanity has ever found that Mars might once have been home to something living. But the strangest part of the whole discovery? This rock isn't even that old. at least not by Martian standards. When scientists plan missions to search for life on Mars, they focus on the oldest rocks they can find. The logic makes perfect sense. Mars had a thick atmosphere 3.8 billion years ago. Liquid water covered parts of the surface, rivers carved valleys, lakes filled craters. The temperature stayed above freezing for long stretches. If life ever took hold anywhere on the red planet, it would have been during that early warm, wet period when conditions resembled a young Earth. Then something catastrophic happened. Mars lost its magnetic field. Without that protective shield, solar wind from our star started stripping away the atmosphere. One molecule at a time. Over millions of years, the air thinned. Surface pressure dropped. Liquid water couldn't stay stable anymore. It either froze into the ground as ice or evaporated into space. The rivers stopped flowing. The lakes dried up. The planet became the frozen desert we see today. Based on that timeline, scientists assumed any Martian life would have died out 3 billion years ago, when the habitable conditions disappeared. So most Mars missions deliberately target the most ancient rock formations, the ones laid down during that early wet era. That's where you'd expect to find biosignatures if they exist anywhere. More recent rocks from after the planetary catastrophe shouldn't contain evidence of life. There wasn't supposed to be anything alive to leave evidence. Cheyava Falls breaks that assumption completely. The Bright Angel Formation isn't among the oldest rocks in Jezero Crater. It's younger. Not by a little, by hundreds of millions of years. It formed during a time when Mars was supposed to be mostly dead already. The atmosphere was thin. Surface water was scarce. Temperatures were plunging toward permanent freeze. Yet here's this rock, packed with organic carbon, sulfur, phosphorus, and minerals that suggest biological activity. Leopard spots that might represent microbial colonies clustering around chemical energy sources. If those spots really are biosignatures, the implications are staggering. It means Mars stayed habitable much longer than we thought. Or it means life found ways to adapt and survive as conditions deteriorated. Maybe it retreated underground where liquid water still existed. Maybe it clustered around geothermal vents that provided warmth and nutrients. Maybe it went dormant during harsh periods and revived when conditions briefly improved. Life on Earth does all of these things. Bacteria survive in Antarctic ice, in boiling acidic springs, miles underground in solid rock. They endure radiation that would kill us instantly. They wait decades in suspended animation for water to return. This changes the entire search strategy. We can't just look at ancient rocks anymore. We need to examine younger formations too. Sedimentary deposits from the late Noachian period, even Hesperian-age terrains which are younger still. If life adapted to Mars's long decline, if it persisted in hidden pockets long after the surface became hostile, then biosignatures could be scattered throughout the geological record, not just in the oldest layers. The Perseverance team realizes something else that makes them pause. Cheyava Falls came from one of the youngest sedimentary formations they've explored so far. It wasn't a primary target. They were supposed to keep moving toward older sites, deeper in the crater. But they stopped, because the rock looked interesting. That single decision to investigate instead of drive past might have just shifted our entire understanding of when and where life could have existed on Mars. And it raises an uncomfortable question that nobody wanted to voice out loud. If life survived this recently in Mars's timeline, what if it never fully went extinct? What if there are still microbes, somewhere below the frozen surface, clinging to existence in pockets of liquid water we haven't found yet? Scientists prefer data over speculation, but sometimes the data pushes you toward conclusions you didn't expect. Vivianite and greigite don't just randomly show up together in rocks. Their presence as a pair tells a very specific story. On Earth, you find vivianite in wetlands where organic material decays slowly underwater. In marshes, around fossilized bones buried in mud. Anywhere dead tissue breaks down in oxygen-poor conditions. It's a hydrated iron phosphate that forms when microbes decompose organic matter and release phosphorus into the surrounding water and sediment. The chemistry is straightforward. Dead cells contain phosphorus in their DNA and membranes. When bacteria break, those cells apart, phosphate ions dissolve into the water. If iron is present and oxygen is low, the phosphate combines with iron to form vivianite crystals, pale blue or white when fresh. The mineral acts like a chemical fossil, marking the spot where organic decomposition happened even after the organic matter itself has completely disappeared. Greigite is rarer and more specific. It's an iron sulfide that certain bacteria produce as a direct byproduct of their metabolism. Sulfate-reducing bacteria take sulfur compounds from their environment and use them as electron acceptors to generate energy. It's a form of respiration that works without oxygen. These bacteria thrive in environments where oxygen is absent but sulfur is abundant. Deep ocean sediments, toxic waste sites, hydrothermal vents. As they metabolize, they excrete greigite as waste. The mineral accumulates around their colonies. Finding both minerals in the same location means you need very specific conditions. You need phosphorus-rich water where organic matter is breaking down. You need iron dissolved in solution. You need sulfur compounds available for bacteria to metabolize. You need oxygen levels low enough that vivianite can form instead of other iron minerals. And you need bacteria actively processing sulfur to produce the greigite. Getting all of these conditions to align isn't simple. It requires a functioning ecosystem with nutrients cycling through it. Mars doesn't have wetlands today. It hasn't had liquid water flowing on the surface for more than 3 billion years. But Neretva Vallis was different back then. It was a river carving through highlands and dumping into Jezero Crater. The water slowed when it reached the lake. Fine sediment settled in delta lobes, spreading across the crater floor. Organic molecules washed in from upstream, where they could have been synthesized by atmospheric chemistry or delivered by meteorites. If microbes existed in that ancient lake, they would have had everything they needed to survive. Carbon for building cells, sulfur and iron for energy metabolism, phosphorus for constructing DNA and membranes. The delta environment would have been calm enough for communities to establish. Nutrients would have concentrated in certain zones where water chemistry favored their accumulation. Microbes would have colonized those zones, fed on the available chemistry, multiplied, and when they died, the minerals would have precipitated out, preserving their metabolic fingerprint in the rock. The reaction fronts in Cheyava Falls capture that exact sequence. Vivianite marking where phosphorus was released. Greigite showing where sulfur metabolism happened. Both minerals locked into leopard spot patterns that suggest organized biological activity rather than random geological processes. The team runs isotope analysis on the minerals. The ratios lean toward biological signatures, not perfectly. There's still some ambiguity, but close enough that dismissing the life hypothesis becomes harder with every test they run. Then they find something that makes the case even stronger. The organic carbon isn't just present in the rock, it's concentrated exactly where those mineral patterns appear. Organic carbon on Mars isn't a new discovery. Curiosity rover found it years ago in Jezero Crater. Meteorites rain down on the planet constantly, delivering organic molecules from space. Atmospheric chemistry can synthesize simple carbon compounds without any biology involved. So when instruments detect organic carbon in Martian rocks, scientists don't automatically assume life created it. The real question is always about context and concentration. The carbon in Cheyava Falls is different from anything detected before. It's not scattered randomly through the stone like you'd expect from meteorite contamination. It's not spread evenly like atmospheric deposition would produce. It's concentrated in specific zones, localized, organized. And those zones correspond exactly to where the leopard spots appear, where vivianite and greigite form their reaction fronts. Perseverance uses an instrument called SHERLOS to find it, scanning habitable environments with Raman and luminescence for organics and chemicals. The name is a forced acronym, but the technology is sophisticated. It fires an ultraviolet laser at the rock surface and measures how molecules scatter the light. Different organic compounds produce different scatter patterns, like a fingerprint for chemistry. SHERLOC can identify what types of carbon-based molecules are present even in tiny concentrations. The signal coming from Cheyava Falls is strong. Too strong to be random contamination, too localized to be from meteorites or atmospheric fallout. The organic carbon sits right next to the iron minerals, right where you'd expect it if all three components came together in the same chemical or biological process. On Earth, that arrangement is called biogeochemical cycling. Microbes consume organic matter. They metabolize it using chemical reactions with iron and sulfur. As they process nutrients, they excrete waste products. Minerals precipitate around the active zones. Everything locks together in the sediment as the rock forms. The pattern in Cheyava Falls matches that Earth process almost exactly. Carbon, iron, sulfur, phosphorus, all sitting together in reaction fronts that mark boundaries between different chemistries. The team runs tests to characterize the carbon more precisely. Is it simple molecules like methane or formaldehyde, or complex chains like amino acids and lipids? SHERLOC can detect organic compounds, but it can't sequence them. It can't tell you their exact molecular structure. That requires laboratory equipment that weighs tons and needs constant power. What the instrument can measure is distribution and association. The carbon molecules cluster with the minerals in ways that suggest they were part of the same formation process. Not added later, not deposited separately, but integral to whatever chemical or biological reactions create the leopard spots. That's compelling evidence, not proof, but another piece of the puzzle that aligns with biology better than it aligns with random geology. The team knows they're building a circumstantial case. No single measurement proves life existed in this rock. But when you stack them together, the alternatives start falling apart. Organic carbon concentrated in reaction zones, minerals that form around decaying matter on Earth, isotope ratios that lean biological, geological context suggesting ancient habitability. Each data point on its own could have non-biological explanations. Together they paint a picture that's hard to ignore. There's just one shadow hanging over the whole investigation. A warning from history about getting too excited too soon. About claiming evidence of life on Mars, and then having to take it back when the scrutiny gets deeper. The team remembers Allen Hills. They remember what happened when scientists moved too fast. August 6th, 1996. NASA holds a press conference at their headquarters in Washington. The room is packed with reporters. Cameras flash. President Bill Clinton makes a live statement from the White House. Scientists have found a meteorite from Mars, designated Allen Hills 84001 after the Antarctic ice field where it was discovered. And inside that 4-pound chunk of rock are structures that might be fossilized bacteria. Microscopic shapes that look like tiny worms. Carbonate minerals that form in liquid water. Organic molecules called polycyclic aromatic hydrocarbons. Magnetite crystals similar to what certain Earth bacteria produce. The announcement makes front-page headlines globally. Life on Mars. Proof that we're not alone in the universe. The scientific paper gets published in Science, one of the top journals. The lead researcher appears on national television. Public interest in space exploration explodes overnight. Funding requests for Mars missions get approved. NASA's budget increases. Everyone wants to know more about these ancient Martian microbes. Then, the scrutiny begins. Other research teams get access to samples from the meteorite. They start running their own tests, looking for confirmation, looking for flaws, and they find problems. Big ones. The microscopic structures that look like fossilized bacteria might just be mineral artifacts. crystal formations that mimic biological shapes without actually being alive. Magnetite and other iron minerals can form through high-temperature processes that have nothing to do with life. The organic molecules might be contamination from Earth. The meteorite sat in Antarctic ice for 13,000 years after landing. Plenty of time for terrestrial organics to seep in. One by one, the biosignature claims start falling apart. Alternative explanations emerge that don't require biology. The temperatures inside the meteorite were probably too high for life. The shapes are too small to be functional cells. The chemistry doesn't match known biological processes as closely as first claimed. By the early 2000s, scientific consensus shifts hard. Allen Hills is not evidence of Martian life. It's a reminder that extraordinary claims need extraordinary evidence. That you can't jump to conclusions based on a few suggestive features that might have other explanations. The backlash is severe. The researchers who made the announcement face criticism for overselling the findings. Public trust in Mars exploration takes a hit. Media coverage turns skeptical. Some of the momentum for Mars mission stalls. The lesson becomes embedded in astrobiology. Be careful. Be thorough. Test every alternative before claiming you found life. Because if you're wrong, the consequences damage the entire field for years. That's why the Perseverance team approached Cheyava Falls so differently. When the initial data came back showing leopard spots and vivianite and greigite, they didn't rush to announce anything. They spent a year testing alternatives. Could high heat explain the minerals? They checked the thermal history. Could extreme pH create the patterns? They looked for acid weathering signatures. Could radiation rearrange the chemistry into these forms? They examined isotope ratios. Every single non-biological explanation they could think of, they tested systematically. And one by one, those explanations didn't fit. The rocks never got hot enough. The pH stayed neutral. The isotopes leaned biological. The geological context supported ancient habitability. It wasn't one or two suggestive features like Allen Hills. It was a whole suite of evidence pointing the same direction. Multiple independent lines of data, all consistent with biological activity. That's the key difference. Allen Hills had ambiguous signals that could go either way. Cheyava Falls has stacking evidence that gets harder to explain without invoking life. The team published in Nature only after surviving peer review from skeptical experts who tried hard to poke holes in the claims. But even with all that caution, there's still only one way to be absolutely certain. You have to bring the rock back to Earth and analyze it with equipment that doesn't fit on a rover. Sapphire Canyon sits inside Perseverance's sample cache right now, sealed in a titanium tube no bigger than a marker pen. Sterile. Protected from radiation and temperature swings. Waiting. It's one of the 27 rock cores the rover has collected since touching down in Jezero Crater back in February 2021. Each sample was chosen deliberately. Each represents a different piece of Martian history. Different rock types, different time periods, different chemical environments. The mission plan was always clear from the beginning. Perseverance collects the samples, stores them. Then a future mission comes to Mars, picks them up, and brings them home to Earth. That's Mars Sample Return, a multi-billion-dollar international collaboration involving NASA, the European Space Agency, and some of the most complex robotics and spacecraft ever designed. 3 separate vehicles working in sequence: a lander to touch down near Perseverance, a fetch rover to collect the sample tubes, and a Mars ascent vehicle to launch them into orbit where another spacecraft catches them and returns to Earth. Here's the problem eating away at the entire program. Mars Sample Return is in serious trouble. Budget cuts, political pressure, competing priorities pulling funding in different directions. The current administration's budget proposal for NASA slashes science funding by nearly half. 41 missions face termination or severe operational reductions. Sample Return is prominently on that list. The vehicles needed to retrieve Perseverance's samples haven't been built yet. The technology exists, but the hardware doesn't. And now there's serious discussion about cancelling the program entirely before construction even starts. Without sample return, Cheyava Falls stays on Mars forever. The leopard spots remain unexplained. The biosignature question stays unanswered. Because as sophisticated as Perseverance's instruments are, they simply can't do what laboratories on Earth can do. They can't run isotopic analysis with precision down to individual atoms. They can't extract organic molecules and sequence them to see if they match biological templates or random chemistry. They can't use electron microscopes to examine mineral structures at nanometer scales, where you could potentially see fossilized cell walls or other biological architecture. All the definitive tests that would prove or disprove the biosignature claims require bringing the rock home. Period. There's no other option. The instruments needed are the size of rooms. They need steady power, constant calibration, consumable reagents. You cannot miniaturize them enough to fit on a rover. Not with current technology. Maybe not ever. The astrobiology community is pushing back hard against the cuts. Articles in Nature and Science arguing why sample return matters. The Planetary Society mobilizing public campaigns. Scientists testifying before congressional committees. Because this isn't about one rock or one mystery. This is about answering the biggest question in all of science. Are we alone in the universe? If life existed on Mars, if those leopard spots genuinely represent ancient biology, then life isn't unique to Earth. It's a cosmic phenomenon. It emerges wherever conditions permit. That fundamentally changes our understanding of our place in reality. And if the mission gets cancelled, if budget politics kills sample return before it launches, we might never know the answer. We'll have spent billions sending rovers to Mars, collected the most important samples in the history of planetary exploration, and then left them sitting in the dirt because we couldn't finish what we started. The clock is ticking. Decisions get made in budget cycles, and every delay makes the mission less likely to survive. When scientists evaluate potential evidence of extraterrestrial life, they use a structured framework called the Confidence of Life Detection Scale, CoLD for short. It runs from level 1 through level 7. 7 distinct stages of increasing certainty. Level 1 sits at the bottom. Detection of a potential biosignature. Something interesting that could possibly be biological but needs more investigation. Level 7 sits at the top. Independent confirmation of life detected beyond Earth. Multiple teams using different methods all reaching the same conclusion. Verified, certain, undeniable. Cheyava Falls currently sits somewhere between level 1 and level 2. More than just a potential signal. More than a single ambiguous feature, but still less than confirmed. Evidence that passes every possible test. And that placement matters enormously for how the discovery gets communicated and how other scientists respond to it. The scale exists because claims of extraterrestrial life have a troubled history. A pattern of initial excitement followed by deflation when scrutiny reveals alternative explanations. The Viking landers touched down on Mars in 1976. They ran experiments that detected chemical reactions in the soil. Reactions that looked biological. That seemed to show metabolism happening in Martian dirt. But further testing revealed the chemistry could be explained by reactive perchlorates in the soil. No biology required. The signals were real. The interpretation was wrong. The Allen Hills meteorite announcement in '96 seemed bulletproof at first. Multiple lines of evidence all pointing toward fossilized bacteria. Then other teams found problems with each piece of evidence, alternatives that didn't need life. The claim collapsed under its own weight. Even the phosphine detection in Venus's atmosphere announced in 2020 remains heavily disputed. Some researchers say it's real and might be biological. Others say the signal could be sulfur dioxide misidentified, or calibration errors in the data. The debate continues unresolved. The CoLD scale forces researchers to be honest about uncertainty. It separates genuine scientific findings from media hype. It acknowledges that discovering life is a process, not a single moment. You don't go from nothing to confirmed in one announcement. You build evidence step by step. You test alternatives. You increase confidence gradually as data accumulates, and alternative explanations get ruled out. Level 1 means you found something worth investigating. An anomaly. A signal that doesn't fit standard geology. Cheyava Falls has multiple signals, leopard spot patterns, vivianite and greigite minerals forming together, organic carbon concentrated in reaction zones, isotope ratios leaning biological. That collection of evidence pushes it toward level 2, the point where the biosignature hypothesis becomes harder to dismiss than it is to accept. But reaching level 3 requires ruling out all plausible abiotic processes. Every single alternative explanation has to fail, and that's brutally difficult. Because even if 99% of the evidence points toward life, that remaining 1% of doubt keeps the claim from being definitive. Science demands certainty before making extraordinary claims, and finding life on another planet is the most extraordinary claim possible. The Perseverance team chooses their language carefully. They don't say we found life. They say this is a potential biosignature warranting further investigation. It frustrates the public who want clear yes or no answers. But it's responsible science. Jumping to conclusions without enough evidence destroys credibility. And if you're wrong, the backlash sets the entire field back for decades. Trust evaporates, funding dries up, future missions get cancelled. So the team stays cautious. They publish the data. They present their interpretations. They acknowledge uncertainty. And they wait for more evidence that could push the confidence level higher. But that evidence can only come from one place: Earth laboratories analyzing the actual rock. A reaction front is a boundary, a line in three-dimensional space where two different chemical environments meet and interact. Imagine groundwater moving slowly through porous rock. The water carries dissolved minerals, iron, calcium, sulfur compounds, maybe organic molecules if they're present in the environment. As the water flows, it encounters layers with different chemistry, different pH, different oxygen levels, different mineral composition. When those two chemistries collide, reactions happen. Elements rearrange, new compounds form, metals come out of solution and precipitate as solid minerals. The boundary between the two zones becomes a site of intense chemical activity. And if microbes are anywhere nearby, they detect that gradient. They move toward it. They position themselves right at the interface, where they can exploit the energy difference. This happens everywhere on Earth where life exists. In soil, where bacteria break down dead plants. The organic matter creates one chemical environment. The surrounding dirt creates another. At the boundary, microbes cluster and harvest energy from the difference. In deep-sea hydrothermal vents, where superheated mineral-rich water meets cold ocean, bacteria coat the vent openings, feeding on hydrogen sulfide and other reduced chemicals. In underground aquifers where oxygen-rich surface water meets oxygen-poor groundwater, iron-oxidizing bacteria form thick biofilms at the mixing zone, pulling energy from the gradient. The pattern repeats constantly. Microbes sitting at reaction fronts, metabolizing whatever chemistry is available, leaving behind mineral deposits that mark their presence long after they're gone. Geologists recognize these structures in ancient rocks. They're diagnostic, reliable indicators that biology was active even when no cells or organic molecules survived to be directly detected. The leopard spots in Cheyava Falls look exactly like terrestrial reaction fronts. Dark centers where greigite concentrated, bright halos where vivianite formed. The minerals aren't scattered randomly through the rock. They're organized, layered in rings, structured in ways that suggest something directed their formation. The team builds computer models of the chemistry. They input the sediment composition from Cheyava Falls, add water with dissolved minerals, include organic carbon, include sulfur. Then they simulate what happens if microbes are present and metabolizing. The model produces minerals precipitating in organized rings. The simulation output matches the real rock almost perfectly. The pattern, the mineral types, the spatial arrangement, everything lines up. Then they run the same model without biology. Just chemistry. Just minerals reacting with water under Mars-like conditions. The results change completely. The minerals form, but they don't organize. They scatter. They mix randomly. They don't create leopard spots. It's not definitive proof. Computer simulations are simplified versions of reality. They make assumptions. They ignore complications. But the match is compelling, because the alternative requires inventing a new chemical process, something nobody's discovered yet, something that can create ordered mineral patterns without any biological input, under conditions that exactly match what existed in ancient Jezero Crater. And despite a year of searching, nobody's found that process. So the question becomes about energy. Where did it come from? What powered the reactions that might have sustained Martian life 3 billion years ago? Life needs energy. Life. That's non-negotiable. Every biological process requires it. Building proteins, replicating DNA, moving molecules across cell membranes, responding to the environment. Without a constant energy source, biochemistry stops. Cells die. On modern Earth, most ecosystems ultimately run on sunlight. Plants and photosynthetic bacteria capture light energy and convert it into chemical form. Everything else feeds on that foundation. Herbivores eat plants. Carnivores eat herbivores. Decomposers break down the dead. Solar energy flows through the entire food web. But early Earth, before oxygen filled the atmosphere, supported ecosystems that didn't rely on sunlight at all, called traces in the rock record. Mars 3 billion years ago had the exact same potential. Jezero Crater held a lake fed by rivers from volcanic highlands. Water flowed over basaltic bedrock, dissolving minerals and creating chemical gradients. Iron leached out of the stone. Sulfur compounds formed from volcanic gases. The chemistry was rich, dynamic. If microbes existed on Mars, they could have tapped into those gradients the same way Earth's chemosynthetic bacteria do. Iron in the water could be oxidized, pulling electrons and releasing energy. Sulfur compounds could be reduced, running metabolism in reverse from what oxygen-breathing organisms do. The energy budget would have been modest, nothing like the abundance photosynthesis provides, but sufficient for a simple microbial ecosystem, enough to establish colonies in favorable locations, enough to persist as long as liquid water remained available. The Bright Angel Formation preserves exactly that kind of chemistry. Sulfur concentrations are elevated throughout the sediment. Iron is abundant in multiple oxidation states. Phosphorus, essential for DNA and cell membranes, appears everywhere. It's a metabolic buffet, all the ingredients laid out and waiting. The leopard spots suggest something used those ingredients. Something clustered at reaction fronts where energy gradients were steepest. Something that fed and metabolized and left chemical fingerprints when it died. But there's a limit to inference from minerals alone. You can reconstruct the chemistry, you can model the energy budget, you can show that conditions were suitable for life. But you can't prove biology happened unless you find the organic molecules themselves. The carbon-based structures that only life produces. Amino acids linked in specific sequences. Lipids forming cell membranes. Nucleotides that encode genetic information. Perseverance detected organic carbon in Cheyava Falls, but it can't sequence that carbon. Can't determine if the molecules are biological or just random hydrocarbons synthesized by violence. Sedimentary rocks tell you about water and life. That's the fundamental difference between them. Volcanic stone forms from molten magma erupting from deep underground. Temperatures reach over 1,000 degrees Celsius. Everything organic burns away. Amino acids break apart. Cell membranes combust. DNA strands shatter into fragments. The heat destroys biological molecules completely. What remains is just chemistry, just minerals crystallizing as the lava cools. Sedimentary rock forms through an entirely different process. Particles settle slowly in water. Clay eroded from distant highlands, silt carried by rivers, sand deposited on lake bottoms, organic molecules washing downstream, dead microbes sinking through the water column. Everything accumulates layer by layer, over thousands of years, over millions of years. The weight of new layers compresses the old ones beneath. Water gets squeezed out, minerals cement the particles together, rock forms, and if conditions stay cool and stable, the organic chemistry survives, gets locked into the stone, preserved for billions of years. Earth's sedimentary record is packed with biosignatures, stromatolites built by ancient cyanobacteria 3.5 billion years ago. Layered structures you can still see in Australian rocks today, microfossils scattered through shale formations. Mineralized cells small enough you need electron microscopes to see them, but unmistakably biological in shape. Isotopic ratios in carbon and sulfur that reveal metabolic pathways. Chemical signatures of photosynthesis, of sulfur reduction, of methane production, all preserved because sedimentary rocks can trap fragile organic chemistry that volcanic rocks obliterate. That's exactly why Perseverance targeted Jezero Crater. It's an ancient lakebed. Sedimentary deposits stacked dozens of meters thick. Different layers representing different time periods in Martian history. Wet periods, dry periods. Chemical changes in the water. If a life ever existed on Mars, if microbes ever colonized that ancient lake, this is where evidence would survive. The rover deliberately drilled cores from mudstone, from siltstone, from fine-grained sandstone, all the rock types that form in calm water where biology could establish and leave traces. Cheyava Falls is mudstone, fine-grained sediment composed mostly of clay minerals. On Earth, mudstone ranks among the best preservers of ancient life. It forms from tiny particles settling in still water, lakes, lagoons, deep ocean floors. The fine grain size means low permeability. Water can't easily flow through and wash away organic molecules. The chemistry stays stable. Biosignatures don't degrade as rapidly as they do in coarser rocks, where groundwater circulates more freely. The leopard spots in Cheyava Falls exist because the mudstone protected them. 3 billion years of cosmic radiation didn't destroy the minerals. Temperature swings between Martian day and night didn't crack them apart. Meteorite impacts nearby didn't shock the chemistry into something unrecognizable. The vivianite and greigite remain. The organic carbon remains. The reaction fronts remain. Still organized. Still telling their story. Still preserved in stone that formed when Mars had flowing rivers and standing water. But reading that story completely requires instruments. Perseverance simply doesn't carry. The rover can detect. It can measure concentrations. It can identify mineral types. What it cannot do is sequence organic molecules. Cannot examine structures at atomic resolution. Cannot run the definitive tests that distinguish biology from geology with absolute certainty. Those tests exist. They're waiting in laboratories on Earth. And the rock that holds the answers is sitting on Mars in a titanium tube. Isotopes are variants of the same element with different weights. Carbon comes in 3 main forms. Carbon-12 has 6 protons and 6 neutrons. Carbon-13 has 6 protons and 7 neutrons, making it slightly heavier. Carbon-14 has 6 protons and 8 neutrons, and it's radioactive. All 3 are carbon, all behave the same chemically, but biology treats them differently. Living organisms prefer lighter isotopes. Enzymes work faster with carbon-12 than carbon-13 because the lighter atom moves more easily through biochemical reactions. It takes less energy to manipulate. So when microbes consume carbon from their environment and build it into cells, they accumulate more carbon-12 and less carbon-13 than the raw materials they started with. The ratio shifts, gets enriched in the light isotope. When you analyze organic material and find an unusually high proportion of carbon-12 compared to carbon-13, it suggests biology was involved in creating that material. The effect is called isotopic fractionation. Life creates an imbalance. Random chemistry doesn't fractionate isotopes as strongly. Abiotic processes produce more even distributions. The carbon-12 to carbon-13 ratio stays closer to what you find in the source environment. So by measuring that ratio precisely, you can distinguish biological carbon from geological carbon. It's one of the most powerful tools in astrobiology. The organic carbon in Cheyava Falls shows isotopic fractionation. Not extreme, not as strong as what you see in modern Earth organisms, but detectable. The ratio leans toward lighter carbon in ways that match biological processes better than purely abiotic chemistry. It's another piece of evidence stacking on top of the minerals and the reaction fronts. Another signal pointing toward life. Sulfur isotopes tell a similar story. Sulfur has 4 stable isotopes. Biology processes them differently depending on how microbes metabolize sulfate or sulfide compounds. Sulfate-reducing bacteria preferentially use lighter sulfur isotopes when they pull energy from sulfur chemistry. The waste products they excrete get enriched in light isotopes, while the unused sulfur in the environment gets enriched in heavy isotopes. The pattern is distinctive, diagnostic of biological sulfur cycling. The greigite mineral in Cheyava Falls carries isotopic signatures that resemble Earth bacteria more closely than abiotic sulfur chemistry. Not perfectly. There's overlap. Volcanic processes can fractionate sulfur isotopes too under certain conditions, but the pattern fits biology, especially when combined with everything else: the vivianite, the organic carbon, the leopard spot organization. Each individual signal is ambiguous. Together they reinforce each other. Here's the frustration though. Perseverance can measure isotope ratios roughly, approximately. The instruments on board give you ballpark numbers, but they lack the precision needed to rule out all alternative explanations definitively. Earth laboratories measure isotopic differences down to parts per thousand. They can separate biological signals from geological noise with high confidence. They can compare Martian samples to terrestrial analogs from similar environments and quantify how closely they match. Those measurements require mass spectrometers the size of refrigerators, instruments that need calibration gases, that need vacuum systems, and precise temperature control. You cannot miniaturize that technology enough to send it to Mars. Not yet. Maybe not for decades. So the isotope data from Cheyava Falls remains suggestive rather than definitive. Compelling but not conclusive. One more reason why bringing the sample back to Earth stopped being optional and became essential. Cheyava Falls didn't form in isolation. It's part of a much larger story written in stone across Jezero Crater. The Bright Angel Formation extends for hundreds of meters along the edges of Neretva Vallis. Multiple rock outcrops, multiple layers, all deposited during the same general period when water still flowed through this region. The rover has been exploring these outcrops for months, drilling samples, taking measurements, building a geological timeline. The valley itself tells you about catastrophic water flow. Neretva Vallis cuts through older terrain like a scar, 400 meters wide at its broadest point, carved by a river moving with tremendous force, enough to carry boulders, enough to strip away layers of rock and deposit them downstream. Geologists estimate the water volume was enormous, comparable to major terrestrial rivers during flood events. This wasn't a gentle stream. This was a torrent. When that torrent reached Jezero Crater, physics took over. The water slowed down as it spread across the wider crater floor. Velocity dropped. Carrying capacity decreased. The heavy particles the river was transporting started settling out. First the boulders, then cobbles, then gravel and sand, finally the finest particles. Clay and silt suspended in the water column. Those settled last, drifting down through calm water and accumulating in delta lobes that spread across the ancient lake bottom. These deltas are geologically critical. They're transitional environments. River meets lake. Fast current meets still water. Energy drops. Particles sort by size. Organic matter accumulates because it settles slowly. On Earth, river deltas are biodiversity hotspots. They're nutrient-rich. They provide multiple microhabitats, shallow zones and deep zones, oxygen-rich zones and oxygen-poor zones. Different organisms specialize in different niches. If Mars ever supported an ecosystem, deltas would have been prime locations for it to develop. Perseverance has been drilling cores from different parts of the Jezero Delta for over 3 years now. Each sample represents a different layer in the geological record. Some are sandstone deposited when the river was flowing fast and carrying coarse material. Others are mudstone from quiet periods when the lake was calm and fine particles could settle undisturbed. Cheyava Falls comes from one of those mudstone layers, fine-grained, rich in clay minerals, chemically balanced. But here's what makes the discovery even more significant. The rock isn't unique. Other outcrops in the Bright Angel Formation show similar features: same white calcium sulfate veins, same mineral composition, same elevated organic carbon concentrations, not identical. Each location has variations, but the pattern repeats. Multiple sites showing similar chemistry, similar geological context, similar potential for preserving biosignatures. That repetition matters. Isolated anomalies can be flukes, random events that don't mean anything. But when you see the same pattern in multiple locations, it suggests something systematic was happening, something widespread. If the leopard spots in Cheyava Falls represent biological activity, then the similar features in nearby rocks suggest that activity wasn't limited to one tiny spot. It was distributed across the formation, across the delta environment, which means if life existed here, it wasn't struggling at the edge of survival. It was established, persistent, widespread enough to leave traces in multiple rock layers across a region. And that possibility changes the entire search for life on Mars. Those white streaks cutting through Cheyava Falls aren't decorative. They're evidence. Calcium sulfate veins that formed when water moved through fractures in the rock long after the original sediment was deposited. Understanding how they formed tells you about the hydrothermal history of this location, about whether liquid water persisted here after the lake dried up, about chemistry that could have sustained life even as surface conditions deteriorated. Here's how the process works. The mudstone forms first. Sediment settles in the lake, compacts under pressure, turns to stone. Then something cracks it. Maybe tectonic stress from the crater adjusting. Maybe temperature changes causing expansion and contraction. Fractures open. Water from elsewhere seeps into those cracks. The water is carrying dissolved minerals. Calcium from weathered basalt. Sulfate from volcanic gases. The minerals stay dissolved as long as the water flows. But when the flow stops or the water evaporates, the chemistry changes. Calcium and sulfate concentrations increase beyond saturation point. Crystals start forming. Calcium sulfate precipitates out of solution and deposits along the fracture walls. Over time, the entire crack fills with white mineral deposits. The vein is born. On Earth, you see these structures in desert environments, in limestone caves, anywhere groundwater has circulated through fractured rock over extended periods. On Mars, calcium sulfate veins indicate persistent liquid water, not necessarily on the surface, but underground, protected from the thin atmosphere, warmed by residual volcanic heat, or insulated by overlying rock. The veins in Cheyava Falls formed after the mudstone solidified, which means water was still present in this region after the lake itself had dried up. Water that could have sustained microbial life even as surface conditions became hostile. The chemistry also matters. Calcium sulfate forms a neutral to slightly alkaline water. If the water is too acidic, the mineral dissolves instead of precipitating. If it's too alkaline, carbonates form instead. The presence of sulfate veins suggests the pH was moderate, balanced, not extreme in either direction. The kind of water chemistry where Earth microbes thrive. Not too harsh, not too corrosive. Some of the leopard spots in Cheyava Falls sit directly adjacent to these white veins. That positioning isn't random. It suggests a connection. The water that formed the veins likely also delivered nutrients to the locations where the reaction fronts developed. If microbes were present, they would have colonized the fractures, fed on the minerals dissolved in the flowing water, clustered along the chemical gradients where water met rock. When the water finally disappeared, the minerals precipitated and locked the biosignatures in place. It's circumstantial. You can't prove cause and effect from spatial association alone. But it's consistent. Everything about this rock fits together. The geological context, the mineralogy, the organic chemistry, the isotope ratios, the reaction front structures. Each piece reinforces the others. Each detail adds weight to the biological interpretation. Still, there's one question the team can't answer with the data they have. Temperature. How hot did this rock get during its history? Because heat destroys evidence. And if Cheyava Falls ever got too hot, everything else stops mattering. When Perseverance first photographed Cheyava Falls from a distance, the images showed an interesting rock. White veins, unusual surface texture, worth investigating. But the cameras at that range couldn't reveal the leopard spots. Couldn't show the fine details that would make this sample the most important find of the entire mission. So the rover moved closer, positioned its instruments, and started high-resolution scans that would change everything. Pixel went first, the planetary instrument for X-ray lithochemistry. It fires a focused X-ray beam at the rock surface and measures what elements are present based on how the atoms fluoresce. The beam is tiny. It can analyze areas smaller than a grain of table salt. Pixels systematically scanned across Cheyava Falls, building an elemental map, pixel by pixel. The data streamed back to Earth over the course of hours. The first thing that appeared was iron, concentrated in small circular regions scattered across the surface. Not random splatters, organized spots, each one rich in iron compounds. Then the scans revealed something unexpected. The iron wasn't uniform. It showed gradients, dark zones in the centers, lighter zones forming halos around them. Chemical variations that suggested the iron existed in different oxidation states. Different minerals, all packed into spots measuring just millimeters across. Next came Sherlock, the Raman spectrometer designed to identify organic molecules and specific minerals. It uses an ultraviolet laser to make molecules vibrate and scatter light in characteristic patterns. Sherlock focused on those iron-rich spots and began its analysis. The minerals came back as vivianite and greigite, both present, both forming distinct layers within the reaction zones. The team went silent when they saw those results. Because on Earth, that specific mineral combination almost always indicates biological activity. The rover zoomed in tighter, maximum magnification. The cameras captured details down to fractions of a millimeter. That's when they saw the ring structures clearly. Not just color variations, actual three-dimensional structures. The spots weren't surface features. They penetrated into the rock. Mineral columns extending downward into the mudstone like pillars. Like something had been growing or accumulating material over time, building up layer by layer. The team ran chemical models. They simulated groundwater moving through this type of sediment, carrying dissolved iron and sulfur, encountering organic carbon. They modeled what would happen if microbes were present and metabolizing. The simulation produced mineral precipitation in organized rings, dark centers where one reaction dominated, bright halos where a different chemistry took over. The output matched the real rock structure almost perfectly. Then they ran the same model without biology. Just chemistry. just minerals and water interacting under Martian conditions. The results changed completely. The minerals formed, but they scattered randomly. No organization, no ring patterns, no leopard spots. The simulations weren't proof. Computer models make assumptions. They simplify complex processes. But the match was striking. Because to explain the observed structures without biology, you'd need to invent a new chemical process. Something that creates ordered mineral deposition in specific patterns under conditions matching ancient Jezero Crater. And despite months of searching, nobody found that process. The imaging revealed one more thing that made the case stronger. The organic carbon wasn't evenly distributed. It was concentrated exactly where the mineral rings appeared. Phosphorus is absolutely essential for life as we know it. It sits at the backbone of DNA and RNA. The double helix structure that stores genetic information contains phosphate groups linking the sugar molecules together. Cell membranes are built from phospholipids, molecules with phosphate heads that organize into barriers separating inside from outside. ATP, the universal energy currency of biology, carries 3 phosphate groups that store and release energy through chemical bonds. Without phosphorus, none of this works. Life cannot function. Earth has abundant phosphorus locked in mineral deposits. Mostly apatite, a calcium phosphate that forms in igneous rocks. Weathering slowly breaks down the apatite and releases phosphate ions into water. Plants and microbes pull those ions from the environment and incorporate them into biological molecules. The phosphorus cycles through ecosystems, from rocks to organisms to soil and back. It's readily available for life to use. Mars has phosphorus too, but not as much, and not as easily accessible. The planet's volcanic rocks contain some phosphate minerals, but Martian weathering is less intense than Earth's, less water, less chemical breakdown. So you wouldn't expect phosphorus to be abundant in sedimentary deposits unless something concentrated it. Yet the Bright Angel Formation is unusually rich in phosphorus, and that phosphorus is locked into vivianite, hydrated iron phosphate, the same mineral that forms around decaying organic matter on Earth. Finding vivianite raises immediate questions. Where did all this phosphorus come from? If it's just geological weathering releasing phosphate from volcanic rocks, you'd expect the element to be spread evenly through the mudstone, diluted, dispersed, but it's not. The phosphorus is concentrated in the leopard spots, right where the reaction fronts formed, right where vivianite and greigite appear together. That localization suggests something was actively accumulating phosphorus in those specific locations. Microbes do exactly that. They pull phosphate from dilute solutions in water and concentrate it inside their cells. They need it for building DNA and membranes. When those cells die and decompose, all that accumulated phosphorus gets released back into the immediate environment. If iron is present in the water, the phosphate combines with it and precipitates as vivianite. The mineral marks the location where biological activity happened, even after the organisms themselves have vanished. The team checked for alternative explanations. Could volcanic activity have delivered concentrated phosphorus to the lake? Possibly. Hydrothermal vents can carry phosphate-rich fluids, but that would spread the element across the entire delta, not concentrate it in millimeter-sized spots. Could specific water chemistry have precipitated phosphorus? In localized zones? Maybe. But there's no known abiotic process that creates the precise spatial pattern observed in Cheyava Falls. Biology provides a mechanism. Microbes cluster around favorable chemical gradients. They harvest nutrients, including phosphorus. They concentrate those nutrients inside their cells at levels far above the surrounding environment. When the cells lyse and break apart, the nutrients dump into the local area. Minerals precipitate. The pattern matches what Perseverance is seeing. Vivianite concentrated in reaction fronts where biological activity could have occurred. It's circumstantial. One more piece of evidence that doesn't prove life on its own. But stacked with everything else, it strengthens the biological interpretation. Each signal reinforces the others. Each measurement points the same direction. And the direction keeps pointing toward ancient Martian microbes that once lived in this rock. Mudstone should be smooth and uniform. That's what happens when microscopic clay particles settle in still water and compress over millions of years. The grains pack together tightly. The texture becomes homogeneous, consistent from one spot to the next. No major variations in grain size or mineral composition. When geologists examine mudstone under magnification, they expect to see a fine-grained matrix, regular, predictable, boring in its consistency. Cheyava Falls looks smooth from a distance. The surface appears typical for mudstone. But when the rover's cameras zoomed to maximum resolution and examined the rock under extreme magnification, the texture changed. Irregularities appeared. The surface wasn't uniform after all. There were tiny pits scattered across certain areas, clusters of crystals breaking the otherwise smooth matrix. Zones where the rock looked almost porous, like something had been there and then dissolved away, leaving voids behind. On Earth, similar textures show up in sedimentary rocks that once hosted microbial mats, biofilms where bacteria form dense communities on surfaces. The organisms secrete sticky polymers that bind them together into structured layers. Living cells mixed with organic glue. These biofilms can be millimeters thick, coating lake bottoms or river rocks. When the biofilm eventually dies and decays, it leaves traces. The organic matrix breaks down. Voids form where cells used to be. Sometimes minerals cast the shapes of the original structures. The rock texture becomes heterogeneous, pitted, uneven in ways that pure sediment accumulation doesn't create. The surface of Cheyava Falls shows exactly that kind of heterogeneity. Not everywhere, just in certain spots. The same spots that contain leopard patterns. The same zones where vivianite and greigite formed, where organic carbon is concentrated. The texture matches what biofilm decay produces. Organic material that was once present broke down and left structural traces in the rock. The team considered alternatives. Could salt crystals have formed in the sediment and then dissolved when conditions changed? That's possible. Evaporate minerals can create porous textures when they precipitate and then get washed away by later water. But there's no evidence of evaporate minerals in Cheyava Falls, no halite, no gypsum in the main rock matrix, just the calcium sulfate in those white veins, which formed much later. Could gas bubbles have been trapped in the wet sediment before it solidified? Gas can create voids, but gas bubbles are random, scattered irregularly through rock. They don't correlate with specific mineral deposits or chemical zones. The pitting in Cheyava Falls isn't random. It's associated with the reaction fronts, with the leopard spots, with locations where chemistry happened. Biofilm decay explains the pattern better than any purely geological process. Microbes forming mats, concentrating phosphorus and other nutrients, creating localized chemical environments where vivianite and greigite could precipitate, then dying and decomposing, leaving behind mineral casts and structural voids that mark where the community once thrived. It's not definitive proof. Texture alone can never prove biology existed. Rocks can be deceptive. Minerals can mimic organic structures through purely abiotic processes. But combined with all the other evidence, the texture becomes one more piece fitting into the larger pattern. One more signal that doesn't contradict the biosignature hypothesis. And the signals keep accumulating faster than the team can explain them away. Temperature history matters enormously when evaluating potential biosignatures. Heat destroys organic molecules. High temperatures scramble isotope ratios. They drive chemical reactions that can create vivianite and greigite through purely abiotic pathways. If Cheyava Falls ever got heated above 200 degrees Celsius for any significant duration, the biosignature claims fall apart. Everything could be explained by thermal chemistry instead of biology. So the team needed to determine how hot this rock got during its 3 billion year history. Certain minerals act as temperature indicators. They form under specific conditions and break down if heated beyond their stability range. Clay minerals in mudstone are particularly useful for this. Smectite clays start losing water and transforming into other minerals above 150 degrees. If you find intact smectite, you know the rock stayed relatively cool. The mudstone in Cheyava Falls contains clay minerals that haven't transformed. That's a positive sign. It suggests temperatures stayed below the threshold where biological molecules would completely combust. The organic carbon is still detectable. That's another good indicator. If the rock had been heated to 400 or 500 degrees, all carbon compounds would have broken down into simpler molecules or burned away entirely. The fact that Sherlock can still detect organized organic chemistry suggests the thermal history was relatively mild. No extreme heating events that would have erased the biosignature completely. But there's a massive gap between what Perseverance can estimate and what the team needs to know with certainty. The rover's instruments can make rough assessments. They can say the rock probably didn't exceed certain temperatures based on mineral assemblages and preserved organics, but probably isn't definitive. There could have been brief heating events, meteorite impacts nearby, volcanic intrusions, hydrothermal pulses. Anything that spiked the temperature for hours or days could have altered chemistry without leaving obvious long-term traces. This is where Earth laboratories become absolutely essential. Scientists have techniques that can measure thermal history with precision. Fluid inclusion analysis examines tiny bubbles of ancient water trapped inside minerals. You heat them in controlled conditions until the bubble expands and pops. The temperature at which that happens tells you the formation temperature. Radiometric dating can reveal if minerals ever reset their isotopic clocks, which indicates heating events. Raman spectroscopy can detect structural changes in carbon that only occur at specific temperatures. These tests require bringing samples back to specialized facilities. You need micro-manipulators to extract fluid inclusions from crystals, mass spectrometers to measure isotopes with extreme precision, all equipment that fills entire rooms and needs constant calibration. None of it can be miniaturized for a Mars rover, not with current technology, probably not for decades, so the temperature question remains partially open. The evidence suggests Cheyava Falls stayed relatively cool. Cool enough to preserve organic chemistry and mineral structures that match biological processes. But there's uncertainty. A gap in the data that non-biological explanations can exploit. As long as that uncertainty exists, absolute proof stays out of reach. The only way to close that gap is sample return. Bring the rock home, run the definitive tests, answer the temperature question conclusively, then move forward with confidence about what the leopard spots really mean. Time is running out to make that happen. When major scientific papers get published, especially ones claiming potential evidence of extraterrestrial life, the response is usually immediate and loud. Critics emerge within days. Alternative explanations flood academic journals. Researchers point out methodological flaws. They propose non-biological processes that could create the same observations. They challenge assumptions. They demand more data. The scientific community tests claims by trying to break them. If a hypothesis survives that scrutiny, it gains credibility. If it collapses under criticism, it gets discarded. The Nature paper on Cheyava Falls, published in September 2025. The scientific community had months to respond. Skeptics could propose competing hypotheses. They could explain how leopard spots form through purely geological processes. They could show that vivianite and greigite appear together in terrestrial rocks through abiotic chemistry. They could challenge the isotope interpretations or point out flaws in the thermal history estimates. Some researchers did exactly that, but not many, and the alternatives they suggested didn't hold up to examination. One early hypothesis proposed that the spots formed through chemical oscillations, a rare phenomenon where minerals precipitate in rhythmic patterns due to feedback loops in solution chemistry. It's been demonstrated in laboratory settings. Certain conditions produce Liesegang rings, concentric bands of precipitated minerals that look somewhat organized. The idea gained brief attention because it offered a non-biological explanation for patterned mineral deposition. But the hypothesis fell apart quickly. Chemical oscillations require very specific conditions: supersaturated solutions, continuous flow maintaining the gradients, and even when those conditions exist, the patterns don't match what Perseverance observed. Liesegang rings are regularly spaced bands. The leopard spots are discrete circular features with internal structure. The chemistry is different. The spatial organization is different. The hypothesis didn't explain why vivianite and greigite would co-occur in the specific ratios found in Cheyava Falls. Another idea suggested impact-induced heating. A meteorite strikes somewhere nearby. The shock wave heats rocks in the surrounding area. Temperatures spike briefly. Chemical reactions accelerate. Minerals precipitate in unusual patterns. Then everything cools before the organic carbon burns away completely. It's theoretically possible. Impacts do cause localized heating. The problem is evidence. There's no sign of an impact in the Bright Angel Formation. No shocked quartz grains that form under extreme pressure. No impact melt. No ejector blankets. No crater nearby that could have been the source. The geology doesn't support the hypothesis. And even if there had been an impact, brief heating doesn't easily explain organized mineral deposition in millimeter-scale reaction fronts. It might create scattered precipitates, random chemistry, not structured patterns. The silence is telling. When biosignature claims are weak or premature, the scientific community responds aggressively. The Allen Hills meteorite faced immediate pushback. Dozens of papers challenged the original interpretation within months. The Vong. That lack of compelling counter-explanation progress, and it makes the case for sample return even more urgent. When NASA announced the Cheyava Falls findings in September 2025, media coverage exploded instantly. Headlines screamed across every platform. Life on Mars ? Ancient microbes discovered ? Biggest breakthrough in space exploration history ? The sensationalism machine went into overdrive. Social media amplified everything. People who had never followed Mars missions suddenly cared deeply. Comment sections filled with excitement and speculation. The story went viral globally within hours. Scientists on the Perseverance team watched with growing dread. Because hype creates backlash. When the public expects definitive proof of alien life and gets potential biosignature instead, they feel deceived, misled. The careful scientific language gets lost in translation. The nuance disappears. Trust erodes between researchers and the people whose tax dollars fund the missions. That erosion has consequences that last for years. The team tried managing expectations from the start. They used precise language in their papers. They emphasized uncertainty repeatedly. They explained the difference between detecting an interesting signal and confirming that life created it. They held press conferences where they carefully outlined what they knew versus what remained unknown. They begged journalists to communicate the limitations alongside the exciting findings. But nuance doesn't generate clicks. Media outlets don't get advertising revenue from headlines that read, "Scientists find something interesting but inconclusive." They need drama. They need certainty. They need stories that grab attention in crowded news feeds. So the careful messaging got distorted, simplified, sensationalized. The public heard life on Mars and tuned out the caveats that followed. They heard ancient microbes and ignored the word potential that preceded it. That distortion creates danger, not just for public trust, but for future funding. When the Allen Hills meteorite turned out to be inconclusive back in the late 90s, public interest in Mars exploration dropped sharply. Budget allocations decreased. planned missions got delayed or cancelled. The enthusiasm that followed the initial announcement transformed into cynicism when the claim didn't hold up. The same pattern could repeat here. If Cheyava Falls eventually turns out to be a false alarm, if some undiscovered abiotic process explains the leopard spots better than biology does, the backlash could destroy Mars sample return before it launches. Public support would evaporate. Politicians would question why they're funding missions that make big claims and then retract them. The entire field of astrobiology could suffer setbacks that take decades to recover from. The scientists know this history intimately. They're walking an impossible tightrope. They need public interest to maintain funding. Mars missions are expensive. They require political support that comes from taxpayer enthusiasm. But too much interest creates expectations science cannot meet. Extraordinary claims demand extraordinary evidence, and the evidence available right now, while compelling, isn't extraordinary yet. It's suggestive, intriguing, worth pursuing, not definitive. So the team publishes in peer-reviewed journals, they present at scientific conferences, they avoid sensational language, they emphasize what they don't know as much as what they do. And they hope the message penetrates through the media filter. That people understand potential biosignature means maybe, not certainly, not proven, just maybe. The public, meanwhile, just wants a simple answer. Yes or no. Is there life on Mars? Scientists can't give that answer yet. Not without bringing the rock home and running tests that Perseverance cannot perform. And that's where everything gets complicated. Perseverance is an engineering marvel. 7 feet tall, 10 feet long, weighs over a ton. It carries cameras that can image details smaller than a grain of sand. Spectrometers that identify minerals and organic molecules from meters away. A drill that can extract pristine cores from solid bedrock. An entire mobile laboratory designed to explore Mars and search for signs of ancient life. The rover represents decades of technological development and billions of dollars in investment, but it doesn't have everything. There are instruments sitting in Earth laboratories right now that would settle the Cheyava Falls debate in days. Equipment so sophisticated it could definitively answer whether those leopard spots represent biology or geology. We just can't bring those instruments to Mars. Not with current technology, maybe not ever. Mass spectrometry tops the list. These instruments separate molecules by weight and identify their exact chemical structure. If the organic carbon in Cheyava Falls consists of amino acids, mass specs would tell you which ones. Glycine, alanine, valine, the building blocks of proteins. If the carbon is simpler, hydrocarbons like methane or formaldehyde, that would show up too. The difference matters enormously. Amino acids in specific sequences suggest biology. Random hydrocarbons suggest abiotic chemistry. Perseverance can detect that organic carbon exists. Sherlock identifies carbon-containing compounds through laser spectroscopy, but it can't sequence them. Can't tell you their molecular structure. That's a critical gap. The difference between maybe biology and definitely biology often comes down to molecular details invisible to rover instruments. Electron microscopy is another crucial missing tool. These instruments can image structures down to nanometer scales. Individual molecules become visible. If fossilized cells exist in Cheyava Falls, electron microscopes would see them. They could distinguish biological shapes from mineral artifacts that coincidentally look organic. They could examine the internal structure of those leopard spots and determine if the organization reflects cellular architecture or just random chemistry. Perseverance's cameras are excellent. They can resolve features down to tens of micrometers. That's impressive for a rover operating on another planet. But biology happens at nanometer scales. Cell walls are nanometers thick. Proteins are nanometers across. You need electron microscopy to see that level of detail. Cameras on Mars simply cannot get there. DNA sequencing represents the ultimate test. Even ancient degraded DNA leaves fragments behind. short sequences, broken pieces. If Martian life ever used nucleic acids to store genetic information, those fragments might still exist in the rock. Sequencing them would be revolutionary. We could compare Martian biochemistry to Earth organisms, determine if life here and life there share a common origin or evolved completely independently, answer fundamental questions about whether biology follows universal patterns or takes radically different forms in different environments. The sequencing equipment required fills entire rooms. It needs steady electrical power, temperature control, consumable reagents that must be replaced constantly. You cannot miniaturize it enough to launch to Mars, not remotely close. The physics and chemistry simply don't allow it with technology available today. All these instruments exist on Earth. They're used routinely in research laboratories. They're proven technology, reliable, accurate, ready to analyze Cheyava Falls the moment the sample arrives. But only if the sample arrives. Only if Mars Sample Return actually launches and successfully brings those tubes home. Without that mission, the most sophisticated instruments humanity has ever built for detecting life will never examine the most important rock we've ever found. If Mars harbors life, even ancient fossilized microbes preserved in rocks, bringing samples back to Earth creates risk. Not primarily to Mars. The bigger concern is protecting Earth. Planetary protection protocols exist specifically to prevent contamination flowing in both directions. We don't want terrestrial microbes hitching rides to Mars and confusing the search for indigenous life. And we absolutely don't want Martian organisms, if they exist, arriving here and causing unforeseen problems. The risk is genuinely low. Mars is brutally hostile to life as we know it. The atmosphere is 95% carbon dioxide. Surface pressure is less than 1% of Earth's. Temperatures swing from minus 100 degrees Celsius at night to barely above freezing during the day. Ultraviolet radiation bombards the surface constantly because Mars lacks a protective ozone layer. Any organisms that evolved that would be adapted to those extreme conditions, cold, dry, high radiation, they almost certainly couldn't survive Earth's dense, wet atmosphere. Much less infect organisms here that evolved in completely different environments. Almost certainly isn't good enough when you're talking about biological contamination. The consequences of getting it wrong could be catastrophic. Even if the probability is vanishingly small, the potential damage demands extreme caution. NASA and international space agencies have strict protocols for sample return. Multiple layers of containment, redundant safety systems, procedures tested exhaustively before any mission launches. The plan involves specialized facilities built specifically for housing extraterrestrial samples. The tubes from Perseverance would arrive sealed. They must survive re-entry without breaching. That means heat shields, parachutes, careful landing in designated zones far from population centers. Once recovered, the samples get transported to a biosafety containment laboratory, level 4 containment, the highest biosafety rating used for the most dangerous pathogens on Earth. Inside that facility, the samples would be opened under sterile conditions, analyzed with instruments that never touch outside air, monitored continuously for any signs of biological activity, growth, metabolism, reproduction, anything suggesting living organisms are present. Only after years of testing with zero indication of viable life would the samples be released for broader scientific study. Even then, portions would remain in permanent containment as a precaution. It's expensive, it's slow, it adds billions to the mission cost, and years to the timeline. But it's absolutely necessary. Because even though the risk is tiny, the ethics demand we treat it seriously. We have a responsibility not to endanger our own biosphere for the sake of scientific curiosity. There's also a deeper question that gets debated in academic circles. Does Mars have intrinsic value we're obligated to preserve? If life exists there, even in dormant or fossilized form, do we have the right to disturb it? Some ethicists argue for a hands-off approach. Leave Mars alone. Don't bring samples back. Study the planet remotely with rovers and orbiters. Respect its potential biological heritage. Others counter that we have a responsibility to search for life wherever it might exist. That knowledge benefits all of humanity. Understanding whether biology is common or rare in the universe is one of the most profound questions we can ask. And we can't answer it by leaving everything untouched. Science requires investigation, careful investigation, responsible investigation, but investigation nonetheless. The debate remains unresolved. There's no clear consensus, but it's becoming more urgent as sample return transitions from theoretical possibility to actual mission planning, or was transitioning, before the budget cuts threatened to kill everything. Cheyava Falls isn't alone in Perseverance's sample collection. The rover has drilled and sealed 26 other cores from locations across Jezero Crater. Each one was chosen deliberately. Each represents a different piece of the geological puzzle, different rock types, different time periods, different chemical environments. Together they map the environmental history of this region over hundreds of millions of years. Some samples are igneous rocks, dark, dense stone that formed from cooling lava billions of years ago. These tell you about volcanic activity, about the composition of Mars's interior, about heat flow and tectonic processes. They don't typically preserve biosignatures because the temperatures during formation destroy organic molecules, but they provide critical context. They bracket the ages of sedimentary layers. They reveal what the planet was like before water appeared. Other samples come from the crater floor where ancient lava flows cooled and fractured, from the delta front where the river first entered the lake and deposited coarse sediment, from fan deposits where debris flows dump material in episodic floods, from carbonate-rich zones that formed when alkaline water evaporated. Each location adds detail to the story of how Jezero Crater evolved. If Mars sample return succeeds, all 27 cores come back to Earth. Not just Cheyava Falls. That comprehensive approach is critical, because finding one potential biosignature is intriguing. Finding multiple biosignatures in different rock types from different time periods becomes compelling evidence. It suggests life wasn't a brief accident. It was established, persistent, adaptable, widespread enough to leave traces in multiple geological settings. The samples also function as controls. If Cheyava Falls shows biosignatures but the volcanic rocks don't, that supports the interpretation. It indicates the signals are specific to sedimentary environments where biology could potentially thrive. If every sample, regardless of type, shows similar organic chemistry and mineral patterns, that weakens the biosignature claim. It suggests a common abiotic process affecting all the rocks, rather than biological activity in select locations. Scientists have already identified several other samples worth prioritizing. A mudstone core from deeper in the delta sequence that contains similar white veins and elevated phosphorus. A carbonate sample that might preserve evidence of water chemistry when the lake was evaporating. A fine-grained sandstone with organic carbon concentrations comparable to Cheyava Falls. Each one could potentially contain biosignatures, or each could help rule them out by showing different chemistry. That's why the mission is called Mars Sample Return Plural. One sample is a data point, an interesting anomaly that raises questions. 27 samples are a dataset. They provide context, comparison, statistical power to distinguish patterns from noise. They let you test hypotheses rigorously instead of speculating based on limited information. The entire suite represents the most valuable collection of extraterrestrial material ever assembled, more significant than the moon rocks brought back by Apollo astronauts, more important than meteorites collected from Antarctica. Because these samples were chosen specifically to answer the biggest question in science: are we alone? And right now they're sitting in sealed tubes attached to a rover on Mars, waiting while political debates and budget battles determine whether they'll ever make it home. Every month of delay increases the risk that they never will. Mars sample return was supposed to launch in 2028. That timeline is already completely unrealistic. The mission architecture is staggeringly complex. 3 separate spacecraft working in coordination across interplanetary distances, each one pushing the boundaries of current technology, each one requiring years of development and testing before launch. First comes the lander. It has to touch down on Mars near Perseverance's location, navigate precisely to avoid rocks and slopes that could tip it over, deploy a fetch rover that drives to Perseverance and collects the sample tubes from the cache, return those tubes to the lander, all autonomously because radio signals take up to 20 minutes each way. You can't control it in real time from Earth. The lander carries the Mars Ascent Vehicle, a rocket designed to launch from the Martian surface. That's never been done before, not by any space agency. The rocket has to lift the sample container into orbit despite Mars's thin atmosphere and 1/3 gravity. It has to achieve precise orbital parameters so the next spacecraft can find it. That spacecraft is the Earth Return Orbiter. It launches separately, arrives at Mars, loiters in orbit waiting for the ascent vehicle. When the samples reach space, the orbiter executes a rendezvous, captures the sample container, then begins the journey back to Earth. A trip lasting years, protecting the samples from radiation and temperature extremes the entire time. Finally, it approaches Earth. The orbiter releases an entry capsule containing the samples. The capsule must survive re-entry at speeds exceeding 11 kilometers per second. Heat shields have to work perfectly. Parachutes have to deploy on schedule. The landing has to happen in a designated safe zone where recovery teams can retrieve the samples immediately. None of this hardware has been built yet. The Mars Ascent Vehicle exists only in design documents. The Fetch rover is in early prototype stages. The orbiter needs technologies that haven't been fully tested in space. Engineers know how to build these systems in theory, but theory and flight-ready hardware are separated by years of fabrication, integration, and validation testing. Launch windows to Mars open every 26 months when Earth and Mars align favorably. Miss a window and you wait over 2 years for the next one. With the current development status, launching in 2028 is impossible. 2030 is optimistic. 2032 is realistic if everything goes perfectly. And things never go perfectly in space missions. Then came the budget proposals. NASA's science funding cut by nearly half. 41 missions facing termination or severe reductions. Sample return explicitly listed among programs under review. The political environment has shifted. Mars exploration competes with other priorities: climate monitoring, exoplanet research, Artemis lunar missions. There's only so much money to allocate. Every dollar spent bringing rocks back from Mars is a dollar not spent elsewhere. Engineering teams watch the calendar with growing anxiety. Every month without funding approval pushes timelines further right. Every delay compounds. A 1-year setback becomes 3 because of orbital mechanics. Budget uncertainty becomes program cancellation because agencies won't commit to long-term development without guaranteed funding. Meanwhile, Perseverance continues exploring, drilling more samples, storing them in the cache, building the collection that might never come home. The clock keeps ticking, and the window for making this happen is closing. If Cheyava Falls is a genuine biosignature, it demolishes the assumptions that have guided Mars exploration for decades. We've been operating under a simple timeline. Mars had a warm, wet period 3.8 billion years ago, when the atmosphere was thick and water flowed freely. Life could have emerged then if conditions permitted. Then the planet lost its magnetic field. Solar wind stripped the atmosphere. Surface water disappeared. Any life that existed went extinct. So we searched the oldest rocks. the ones from that brief habitable window. Cheyava Falls rewrites that story completely. The bright, Angel Formation is younger, significantly younger. It formed during a time when Mars was supposed to be cold, dry, and dead. Yet here's a rock, packed with organic carbon and minerals that suggest biological activity. If those leopard spots genuinely represent ancient microbes, it means Mars stayed habitable much longer than anyone thought, or it means life found ways to persist as conditions deteriorated, retreating underground, clustering around geothermal vents, going dormant during harsh periods and reviving when water briefly returned. That changes everything about search strategy. We can't just target ancient formations anymore. We need to examine younger rocks too. Sedimentary deposits from the late Noachian period, maybe even Hesperian age terrains which are younger still. If life adapted to planetary decline, if it survived in protected niches long after the surface became uninhabitable, then biosignatures could be scattered throughout Mars's geological record, not concentrated in one narrow time window. It also forces us to reconsider what counts as habitable. Mars lost its magnetic field early in its history. The atmosphere got stripped away, but subsurface water could have persisted, protected by overlying rock, heated by residual volcanic activity or geothermal gradients. Microbes don't need sunlight. They just need liquid water, chemical nutrients, and an energy source. Mars might have provided all three hidden below the frozen surface for billions of years after the planet looked dead from orbit. The implications extend far beyond Mars. If life can survive in marginal conditions, if it can adapt to planetary catastrophe and hang on in refugees, then habitability is more robust than we assumed. Icy moons like Europa and Enceladus become even more compelling targets. They have subsurface oceans beneath frozen shells, protected water that could harbor ecosystems we'd never detect from the surface. Exoplanets that seem too cold or too dry might still host biology in protected underground aquifers. The search for life expands from planets exactly like Earth to anywhere liquid water and chemistry exist. That's a fundamental shift. It means potentially habitable environments are far more common than we thought. It means we should be looking in places we previously dismissed as too extreme. It means the question of whether we're alone might have a very different answer than we expected. That's the real significance of Cheyava Falls, not just that it might be evidence of ancient Martian life, but that it forces us to rethink where life can exist, how long it can endure, how adaptable biology really is when faced with planetary scale disasters. And it all depends on whether we bring that one rock home, whether we complete the mission, whether we answer the question it's asking. The clock is running. The decision point is now. And what we choose will echo for generations.